Hola geeks, welcome to another video. So today we will be discussing about the techniques in biology, and specifically in that we are going to do the technique EMSA. Now this playlist of techniques in biology deal with the questions asked in the part C for CSIR net and through analytical questions in Gate. I have made videos on flow cytometry analysis, that is FACS. I've also made videos on the knockout mice and I have solved problems related to those. We have also done protein purification techniques. We have discussed about ion exchange chromatography, size exclusion chromatography, etc. So if you are interested in any of these videos, do check it out. I'll link it up in the I button or down in the description below. And without much further ado, let's get right into this video. So for today, we are going to discuss the technique EMSA. So what does EMSA actually stand for? It stands for Electrophoretic Mobility Shift Assay. Okay. So simple full form, Electrophoretic Mobility Shift Assay. The name itself gives a lot about the technique. As you can see that the technique has to do something with the electrophoresis. So we are going to electrophorese some DNA or protein. Let's see what it is. And we are going to observe the shift in motility. Now, before even trying to understand how to solve questions, we need to understand the principle behind the EMSA and when to do EMSA. So what EMSA will tell you? So EMSA essentially tells you if a protein interacts with DNA. If you want to find out a protein is interacting with the DNA or not, you go for EMSA. This is an in vitro technique. Now, I can give you a very good example of an instance where you should do ELISA. Like when we discuss processes like replication, transcription, translation, what we say that on the DNA at the say -35 or -10 a protein binds, right? We say that. So who told you this protein is binding to that particular part on DNA? How are you going to explain, okay, that this protein is definitely binding to DNA? So one approach to doing this explanation is carrying out the EMSA technique. Now, after you come to know when you should do EMSA, we should see what is the principle behind EMSA. So you will first discuss a case 1. Now, this is a gel, okay, electrophoresis gel, and these are the wells over here, okay. Now, this end is connected to the negative electrode and this end is connected to a positive electrode, okay. So say now you want to check if the protein A binds to DNA. You want to check this. So you have a DNA and let's say this is the protein A. You want to determine if this protein A actually binds to DNA or not. So first thing what we do is in the lane 1, I will just add the DNA, only DNA. So obviously DNA itself is negatively charged. So it is going to be attracted to the positive electron and it is going to travel in this distance, right? So DNA will travel and let's say the DNA travels to this point. You get a band over here. So we say that yes, this DNA is there and it is traveling up to this particular point. Now, what I do is to this DNA that is under study, I add the protein A. So in vitro conditions, I will take a test tube, I'll add the DNA, I'll add the protein. Okay. Now, I will take this mixture and I will load it in the well. Okay, so let's say the lane 2, okay, is where I have loaded the DNA and the protein. Now, let us consider a case, that is, this is a case 1 where the DNA is actually bound by this protein A. So protein A does bind to the DNA. Let us consider this case. Now, if the protein binds to the DNA, don't you think that this DNA is going to become more heavy? Imagine I ask you to run from point A to point B. You'll run with full speed. Say you reached over here. Okay, this is the point B. Now, what I do is this time I give 2 heavy bags in your hand. 10 kgs each. And now I ask you to run. Will you be able to run this far? Maybe because of that bag you got heavier and you can't run more faster. So what does this mean? When something is attached to you, you are running slower. Similar thing happens with the DNA. If the protein has bound to the DNA, the mobility, okay, the mobility of the DNA will decrease. And therefore, you will observe a band slightly higher than when the DNA is run alone. So can you see there is a shift in the band when you electrophorese this DNA? And this shift is because the reduced mobility of DNA due to binding of protein. Therefore, this technique is called as electrophoretic shift assay. Now consider the case 2. Case 2 is when the DNA doesn't interact with the protein A, or the protein A doesn't interact with the DNA. So in that case, what you will find in the lane 3, when there is no interaction, DNA will run its own distance that it was running usually, and you will get another band of protein A. So somewhere here, according to the molecular weight of that protein, you will get 2 different bands because it is not interacting only. So using this technique, you will be able to predict if a certain protein binds to a DNA or not. Clear? Now the concept will get even more clear when we solve a CSIR net question. Till here, if you are understanding, you're liking the video, please do subscribe to my channel for more videos like this, and comment down below with a thumbs up, or let me know what techniques I should do further. Now, let us move to this question. Okay. So what this question says, a researcher was working with 3 proteins. Okay, he was working with 3 proteins. The proteins are protein A, protein B, and C that have roles in gene expression. In order to validate the hypothesis, he performed EMSA. The purified proteins were allowed to interact with the labeled DNA. So they incubated these 2 and they did autoradiography. Why is this possible? Because they had labeled the DNA. So definitely to observe bands, they did this method. Fine. So now you have to see which of these interpretations can be made and answer the questions properly. So let us look at this autoradiograph. Now, first I want to try and explain to you what is in each lane, and then we'll go from there. So this is the first lane. Okay. In this lane, they did not add protein A, did not add protein B, did not add protein C. Then what did they add? Yes, only the DNA. And when there's only DNA, it travels to this distance, right? Okay. Now, what they are doing in the lane 2 is they are adding the DNA and protein B. What do they observe? See, the results are very similar to the lane 2. What does this mean? This means that the protein B is not binding to DNA. That's why you cannot see it over here. So I can just say that protein B does not interact, right, with the DNA. What does that mean? That essentially means that it doesn't have a DNA binding domain. So look at one of the options. It says protein B possesses a DNA binding motif. Absolutely incorrect. If it had the motif, then it should have bound and the mobility should have decreased. You should have observed a shift in that. Let us move to the lane 3. Similar type of thing is done. Here you add protein C, and on adding protein C also same result is obtained. So that also means that protein C does not have a DNA binding domain. Let's look at the lane 4. Now there is something different we can see. When they added only protein A and the DNA, they observed a shift in band. See, regularly used to get only one band here, but here there is a shift in the band. There is a decreased mobility. That definitely means that protein A has a DNA binding motif, right? and that is the option A over here. So this is a correct interpretation. So where you don't have option A, that won't be your answer. So you can eliminate two options. Now all we need to do is C, C and D options. So let us look at that. Now lane 5 is interesting. In the lane 5, they have only added the proteins A and B. Now, again, you're going to see something interesting. What interesting? Since in both the lane 4 and 5, you had the protein A and DNA, both places you are seeing that interaction, that yes, because DNA A has a binding domain for the protein, for the DNA, that protein has the binding domain, it is binding and therefore the mobility is reduced here. But we are obtaining another additional band, and this band seems to be even more less mobile than the previous, right? So what we can say is when along with A you have B also present, the mobility has decreased further. Why it may be? See, this is the DNA, this was the protein A. Now, because you're getting another band with further reduced mobility, that means the DNA became more heavy. How can you explain this? Exactly. This protein B must be now binding to this protein DNA complex. Correct? So that is an option. Protein B binds to DNA protein A complex. For just understanding purpose, let's discuss the lane 7. Lane 6 is very similar to 1, 2, 3. So I'm not discussing. Lane 7 shows that they added A and C. Now see, when they added both A and C, you can only see the band that corresponds to the protein A complex. What does that mean? That A and C don't interact only. So therefore, this is also not our option. This is incorrect. Thus, A and C are correct, which is option B. So option B is my answer. Hopefully, this helped clear your doubts about this topic. If you have any other questions, please let me know in the comments and subscribe to my channel. I'll see you in my next video. Bye.